Good morning and good afternoon. Thank you for joining day 3 of our 2020 virtual user meeting, ligand-gated ion channels in drug discovery. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's presentations. By default, all participants are muted upon completion of the presentations. I will open the floor to some questions. We encourage you to submit your questions throughout the course of each presentation. It is now my pleasure to introduce Professor Dr. Stefan Mergler from Charité in Berlin, presenting TRP channels in human corneal cells and their role in ophthalmology. I will now hand over the talk to Professor Mergler.
Yeah, hello everyone. My name is Stefan Mergler. I'm working in the ophthalmology at Charité University, and I would like to introduce you or to present you some interesting findings regarding using the planar patch clamp technology in experimental ophthalmology. On the screen on the left side, you can see a table of contents. So what I want to show you or present you today, on the left side, first introduction. I would first give you an idea how it looks like inside the eye, to remember you the anatomy of the eye, so that you can have an impression what what kind of research is specialized. And then I will also show you an example from some eye diseases and why TRPs are so interesting. And I would like to also introduce you some projects which I am currently working on. Excuse me, my English is not very fluent. Hopefully you can understand me, and I ask for understanding. Okay, then the next part are the methods. You already know the methods, it is known, but I will also highlight some interesting aspects which I have considered in my projects. And then I will give you some examples, thermo-TRPs in cornea cells and also in ocular tumor cells, and I will focus on TRP1, why the role of TRP1 is important at that point. And then I will give you an example, a clinical aspect, dry eye syndrome, for example. I will explain you the problem and results and future directions. At the end, I will give you a summary. Okay, yeah, the first, let me see, is it working? Yeah, the first you can see, this is the anatomy of the inner eye. You can see here, this is the eye can be divided in an anterior part and the back part. As you know, there's retina and the nerve fibers and are connected here in the retina, but also in the cornea. It's not shown here directly. There are also nerve fibers in the cornea, as you know. And the cornea here is a square, the anatomy of the cornea. You can see the epithelium, and then the Bowman membrane is in between the stroma and the epithelium. The epithelium has around 200 layers, and the stroma is 90% of the cornea is stroma or keratocytes including. And then the descendant membrane is coming, and then the last layer is the most important one, the endothelium. The endothelium is only one cell layer which is responsible for pump function, barrier function, and it's totally important for the cornea, especially in the context with cornea transplantation. This is only for your information how the cornea is this interesting part here. So coming to the next, then I would like to introduce some eye disease here. Another human eye anatomy, and you can see again cornea here in the front part and the back part, the retina and so on. And the most, one of the most regular eye diseases is the so-called dry eye syndrome. I will explain you later in an example. And therapy is questioned. So far there's only replacing the fluid by artificial fluid to get against symptoms, and eye drops are regularly used. I will come to that later, to that point, what is the current state of research and what is the future direction in that connection. Another interesting example is the so-called tederium. This is a benign tumor which is growing on the surface on the eye. Mostly people who are getting a lot of sunlight, as people living in countries where a lot of sunlight is, can get such a kind of cell overgrowth. The only option is to make a surgery removal, but one of my projects I'm focusing on investigation of such interesting tissue, what's different between healthy conjunctiva cells and these tumor-like conjunctiva or growing cells. These are only some examples of the eye diseases which may occur. For your information, each of this specific tissue, there are a lot of tissue specialized in the eye. Each tissue can fail, and then each tissue, for example, if the lens is failing, then we have the gray, the lens, what's the English, opacity, lens is not properly working. If there we have a problem with the retina degeneration, for example, macular degeneration, there are so many diseases which are linked with retina. Also with the optic nerve, if the optic nerve is degeneration, they have the so-called glaucoma. And so you cannot imagine how many diseases are existing in the eye. And also the eye surface can also get bacteria or virus, or then the cornea is failing, the light cannot go through the cornea and the lens. This is only example. I would like to focus on the cornea especially, and also some ocular tumors and the projects. As you know, calcium is one of the major second messenger in the cell. There's nearly nothing which is not dependent on calcium. This I want to highlight in this simple figure, showing or demonstrating that characteristics of cells are dependent on calcium signaling. And the calcium regulation is predominantly regulated by calcium channels, of course, including the TRP channels. And here's again the cornea, what is also state of the art. Here again, there's a cross section of the cornea, and inside the cornea there are so-called nerve fibers or stromal nerve bundles. Here's a view how it looks like from the front. Look, the nerves are going radial. And for your information, the pain receptor density is about 7,000 receptors per square millimeter. There's no other tissue in the human body which has such a high receptor density. In other words, the cornea is very, very sensitive. For your information, the nerve fibers are in between the cornea. And here again, the cornea endothelium, and is the cornea epithelium, and in between is the stroma. And in the nerve fibers, it's known from the group in Spain, Alicante, Belmonte et al. They firstly detected TRPV1 channels into the MATE channels in the cornea nerve fibers. This is the classical function of the TRP channels as a pain receptor, of course. And in addition, my collaboration with Peter Reiner, a long-lasting colleague working at the State University of New York, we together detected for the first time TRPV1 in epithelial cells. And TRPV1 has other function, not a classical function in the cornea epithelium. And you can see TRPV1, 3, 4 was detected. And interestingly, also G-protein coupled receptor like cannabinoid receptor, which we also detected for the first time in cornea epithelial cells. And also TRPM8, which is known as a cold receptor, and TRPC4 channel, known as a staurovoid channel. These channels are also expressed in the cornea epithelium. And some 10 years ago, the function of these channels were unknown. And furthermore, in the stroma, 2 years ago, we published a paper. Also, we could detect TRPV1 as well as TRPM8 channels also in the stroma cells and the non-excitable cells, for your information. And finally, it was 2010, the first paper which we published about expression of thermosensitive receptors also in the cornea endothelium, TRPV1, TRPM8, and recently we are just preparing a publication about the function of TRPA1 in the cornea. For your information, the cornea is here, the upper part, and the inner part is here. And the first is like skin. Only the difference is that it's transparent skin, like transparent skin, very complicated. And there's also these chip channels can be activated by temperature change. Therefore, temperature plays a role in this context in the physiology. Okay, coming to the projects. One of the projects, the first project, is focusing on the cornea endothelial cells. And the clinical aspect is regarding investigation, how we can improve storage of donor corneas. Here's an example of a donor cornea for corneal transplantation. For your information, the cornea is the most transplanted tissue known in the ophthalmology and the medicine. And here's an example showing the expression of tryp channels, especially TRPV1 heat-activated channels. Here's red trace showing the activation, heat stimulation, and after stimulation recovery is visible here. This is only a demonstration of heat-sensitive channels for the first time, the cornea endothelial cells. It is also important to know to better understand the physiology of the cornea endothelium, as I said, regarding pump function and barrier function. Therefore, these channels are interesting in this context. We want to know, or my investigations are focusing on the role of tryp channels in the cornea endothelium and how we can improve cell vitality, because classical in a cornea bank, the doctors checking the cornea endothelial cell density and the apoptotic cells. They do some simple stainings checking for apoptotic cells, and they check the density. These are the only two parameters, by the way. And I am just using with the Nanion technology, is very interesting technique for doing a high throughput measurement to assess the quality of donor corneas in aspects regarding the channel activity. And we know there are some publications demonstrating that apoptosis and tryp channels, there's an association. This is one project. Another project is focusing on the eye surface cells, especially cornea epithelial cells. Here's regarding the dry eye. I will come back to that later. This is the first publication which showed in details the electrophysiological properties of the cells in connection. Here you can see we use gluconate instead of chloride to avoid anion channels. And here's a protocol. It's a typical protocol for tryp channel activation, depolarization from -60 to +130 millivolts. This is under control condition, and this is an after application of capsaicin, which is a known TRPV1 channel agonist. As you know, here's including some leak terms, but nevertheless, we can see clearly outward rectifying currents and some inward rectifying currents, which is typical for tryp-like channel current. And here's the under control condition. We can see no outward currents. And that here's again, this is another recording. We're using a ramp protocol, and you can see nearly same, still some leak current, but in the main take-home messages that we can see, nevertheless, some clearly outwardly rectifying currents. This is a project I will explain you on the last here in the dry eye syndrome more details. Only for your information, this is a historical figure showing the TRPV1 channel in non-excitable cornea epithelial cells for the first time. Another project is also focusing on the cornea surface, and I also have a collaboration with some endocrinologists. And Professor Curler called me and asked me, can you please check the effect of tyronomine on your cells? Because we have some experiments with animals showing that there's a hypothermia. That means it has something to do with the temperature. And then we checked, indeed, the tyronomine indeed has an effect on the intracellular calcium regulation. And I would like to show it in this example. Tyronomine is a hormone derivative of the thyroid, here from the thyroid as sitting here. Interestingly, it has some effect in the brain, as it is known in the literature, but so far there are no investigations about connection to tryp channels. And some previous publications we could show an association between tyronomine and TRPM8, interestingly. And this is like a cooling agent. It means if we apply tyronomine to the cells, there's an activation of TRPM8 channels and corresponding calcium influx. And this experiment Here's a cell showing control cones as shown here. There are some cones which may be partially choroid channels or so, but in that context, not so important. And then if here's a, this is a calcium imaging recording, you can see the fluorescence ratio here, and there's a time. And here's a corresponding patch clamp recording. Here's showing the current density, and here's a time course. And this is the time point A on the control condition. And after application of tylonomine, and this induced the clear activation of chip-like channel cones. We have checked this with or chip M8 overexpressed cells and so on, and with a lot of blockers, clearly indicating that there's an association to chip M8. You can see the clearly outward rectifying cones after application of capsaicin. And this is, of course, this point of time here. And of course, there's a following calcium influx, which is shown at that point of time. That means this is like isolene or menthol activating TRPM8 channels. And we cannot exclude also TRPA1 channel activity, which we need further investigation to check this fact. And you can see that there's an increase of calcium. And afterwards, if we applicate capsaicin, in addition to activate TRPV1, and we could see this is here's a control experiment without pre-incubation of tylonomine. There's a clear capsaicin effect as shown here. There's a positive control showing that there's clearly TRPV1 or capsaicin-induced TRPV1 activation in corneal epithelial cells. And in cells pre-incubated with tylonomine, there's no effect visible after application of cap, which is shown here in the time point here. You can see there's no effect, completely abolished, meaning that there's an interplay between TRPM8 and TRPV1. In other words, if we activate TRPM8, TRPV1 cannot be activated. There's a negative feedback indicating that we can modulate the channels by using this interesting tylonomine, which can be also tested in eye drops to avoid capsaicin activation of TRPV1 in cornea cells, which I show you later in the dry eye syndrome explanations. Another project is focusing on eye tumor cells. This is also an interesting topic, because I worked partially also in the gastroenterology, working with neuroendocrine cells. Here, uveal melanoma cells is the most occurring eye tumor in adult. Retinoblastoma is a very aggressive tumor, which is has initially is in children. And neuroendocrine tumor is a rare tumor which can occur in endocrine tumors and endocrine cells in the pancreas, for example, or the ileum. TQM I already explained, this is a benign tumor. And this was a project which also was supported by the GFG. We investigated the role of tape channels in different kind of tumor cells. Here's an example showing retinoblastoma cells. This is a preparation for the planar patch clamp recording. You can see single cells, very small cells. And here is the imaging, colored with red. And here you can see some region of interest which can be investigated for calcium imaging, so that we can see how the calcium is moving. And here this is the same as here is a high magnification. This is a drop for the planar patch. Can be checked before we do the planar patch. Can we check the drop whether the cell density is sufficient? And here's another original calcium imaging showing the typical structure of these retinoblastoma cells. And here this is the recording showing an effect of vitamin C, ascorbinic acid. Interesting, it has an effect on calcium regulation as well on whole cell cones. You can see ABC again, that we use N-acetylcysteine, which is a blocker, also blocking the ascorbinic acid receptor, as well as some ion channels. And you can clearly see an inhibitory effect of that drug, showing that there is indeed an effect on whole cell counts and corresponding calcium regulation. I don't want to go into details to this interesting project. We have very interesting findings, which was just published 2 or 3 weeks ago, and that from my doctoral student from Poland, who did the most interesting experiment in this project. Another project, this is only a feasibility study in the beginning. It's not supported so far as a cooperation with the Charité gastroenterology. And we try to investigate so-called tumor organoids, which are 3D cell culture investigations. And kindly a colleague of the group in the gastroenterology provided some cells. She prepared a drop of this cell suspension here. It was too low. We could not record because the density was too low, but this density was sufficient, and we could, these are only control counts, only checking whether we can record, whether it's feasible to record these cells. Interestingly, it could be recorded. As you can see here, there's a RAM protocol from -60 to +130 millivolts. These are the corresponding answers, and there's a time course showing constant outward currents and inward currents. And here's again a RAM protocol showing current voltage relationship, which also shows typically outward rectifying currents. Mainly, also tube channels can be involved in this interesting study. And the last project, which was started just a few weeks ago, it's a collaboration with the Robert Koch Institute, and they are interested to check for the calcium regulation in Neospora and Toxoplasmosis gondii. So far, we started with Neospora, which is not toxic. And Toxoplasmosis, as you know, perhaps, is every second person is infected with such a Toxoplasmosis gondii. It can be dangerous, cannot be dangerous, depending on several clinical aspects. And the only idea was from my colleagues in the ophthalmology, Professor Plyer, and also the colleague Frank Seber from the Robert Koch Institute, they suggested to checking whether it can be measured with using the planar patch clamp technique. And for the first time, I only did the screenshot some minutes ago because I was in a little bit time pressure to show the latest recording. And the problem is with the preparation of this. Here's an electronic microscopic image showing similar like what's in English? Erreger, I don't know the English. This type of infectious cell type. And the problem is that the preparation is difficult because there's a lot of cell debris. These objects are normally inside a cell, and we need to take them out of the cells. Therefore, the colleague of the doctoral student of the Robert Koch Institute, she prepared the cell suspension by using some scratches to open damage the cells so that these objects can go outside, and then a centrifugation, but they try to optimize the cell suspension preparation because only 1 from 3 recurrences were successful. We try to improve the preparation of the cell suspension of these objects to do more recordings. This is only a simple recording, the same as I showed you before. This is a protocol -60 to +130 millivolt at a holding potential of 0 millivolt. The pattern is different from the others which we measured before. There's some, as you can see in the regarding the inward counts, there's a component which is a little bit non-linear, as you've shown here. So far, we don't know which channels are involved in this whole cell record. It is only first recording only for checking whether it's feasible or not. And so far, it seems feasible if we improve the cell suspension or the preparation for the patch clamping, for the planar patch clamping, as you know. Okay, these are the projects. And now coming to the next point, we are mainly using cell cultures. especially established cell models. For the investigation of corneal endothelial cells, we used a cell model named HTEC12, which was established, I think, 20 years ago by Engelmann, Bednarz, and Ms. Monika Walding. I still have contact to Monika Walding. She's a very good expert regarding corneal endothelial cell research. She is in Dresden, and she kindly provided me these cells for these investigations. And we have checked primary cell cultures and this cell line, and we could see there are similar electrophysiological characteristics indicating this cell line is somehow stable. Oh, sorry, I got a moment. I can come back here. And this another cell line, corneal epithelial cells. It was initially established by a Japan group, and this cell line is used worldwide in the US, in Germany, and on many countries are using this cell line because also established and safe cell line for investigation of corneal epithelial cells. And recently, I think 5 or 8 years ago, Ms. Sohn Krupper from the Eppendorf Hospital in Hamburg established recently human corneal keratocytes, which is specialized fibroblasts of the stroma, and this is helpful for investigation of stroma cells. And these Tegera cells are currently provided from the clinics by Professor Plyer. And then for the investigation of uveal melanoma, we use the so-called uveal melanoma cell line, which was kindly provided by Jaag et al. She's one of the leading investigators regarding uveal melanoma. Very, very interesting. She had beautiful review articles about investigation of uveal melanoma, which is really a challenge for understanding the mechanism. And finally, we investigate retinoblastoma cells. We have 2 cell lines. One cell line is a normal tumor cell line, and the other cell line is a cytostatic resistant cell line. We could see some interesting difference regarding tryptophan channel expression. In other words, regarding the last 2 tumor cell lines, we could clearly show that the progression of these tumors are also dependent on the expression of TRP channels, the gene expression as well as the functional expression of this cells, TRP channels. Okay, these are the cell cultures, only an example. And this is an example showing imaging. We can see here region of interest, and this is an example for a very high, very strong invert, calcium influx. This is an example showing corneal endothelial cells which are overexpressed with TRPV4 channel, kindly provided by Monika's lab in Dresden. And we could see, of course, of the overexpression clearly shows such a strong in calcium influx. Here's an image of the lab. We have also a classical patch clamp setup, but in the moment I prefer to use, of course, the Nanion technology. And by the way, one student asked me, is there a difference between the classical recordings and the planar patch clamping? And in those days, we investigated insulin-producing cells because of the collaboration with the gastroenterologist. And we could see the patterns are exactly the same, of course, but the current density are a little bit different because it depends on the surface of the cell. Because in the classical recording, as you know, the cells are on cover slips. And in the Nanion, we have cell suspension. Therefore, the surface is a little bit different. And for the normalization of the currents by current density dividing to the capacity, cell membrane capacity, the difference may be because of the different surface maybe. But I don't know the exact reason for that, but this is also an indication. Okay, coming to the next, I'm focusing on the temperature sensitive TRPs because in my view, the temperature is a major environmental factor for physiology and function of tissues, especially of the eye surface. And here's a summary showing as you probably know, the temperature range of these channels. And you can see the heat channels here and the cold receptors here on the other side. in between the TRPV4 and TRPV3 channel. And as you know, there are specific kind of drugs or nature compounds which can activate these channels. And in the meantime, I would say last 5 or 10 years, a lot of companies focusing on TIP channel research, developing new tools for investigation of properties of TIP channels. Fortunately, in those days, in the beginning, 20 years ago, when TIP channels are described for the first time, there were less tools for investigation compared to voltage operated calcium channels. There are a lot of tools available, for example, Baike for L-type channels or conotoxin for other channels. For the TIP channels was a little bit less tools available, but so far in the meantime, there are a lot of very high sensitive drugs developed by some companies. Okay, focusing TRPV1. TRPV1 has a lot of factors for activation, especially for heat, as you know, capsaicin, the intercellular binding site, hyperosmolality, which plays a major role in dry, and also the pH value, a change of pH value can also activate TRPV1. And as you can see, is a non-selective cation channel, and on the inner part, also NADA can also activate the channels and inhibit. And also, this channel is also partially voltage dependent, as shown by Nelius et al. Or Clapham showed that this channel can be also in part activated by a voltage change. This is a summary of the different kind of activation mechanism of this channel. Okay, the TRPV1, would like to show an example with the overexpressed TRPV1 transfected human embryonic kidney cell. As you know, there's a cell line which is regularly used in research. And here's again step protocol from -60 to +130 millivolts. And here's the control currents. Probably some of these currents are channels which are active at room temperature, or we can exclude also anion channels like chloride channels. But this is minor in my view. And after application of capsaicin is going clearly outward rectifying currents, and we can also see an increase of invert currents. The invert currents are mainly charged by calcium. This is meaning that the invert currents is corresponding to the calcium imaging. Because we have external solution contains calcium and internal solution is calcium free. Therefore, we have a strong electrochemical gradient from outside to inside. And these invert currents are mainly charged by calcium. Therefore, these currents are interesting in connection with calcium regulation. And outward currents also maybe magnesium or other divalent cations are these outward rectifying currents. And I have developed a more precise method for investigation. 2003, I had published a paper about showing the three-dimensional view of the activity of the channel. The same recording as before. This is under control condition. And here is after a capsaicin application, you can see here the current density. Here's the time. And here's the voltage, so that you can see three parameters together. And if you view on this contour plot, this is shown here, this can be analyzed regarding activation threshold, for example, and you can distinguish between specific and unspecific activation of channels. And we could show for the first time different patterns regarding unspecific blockers, for example, lanthanum or other unspecific nickel chloride, for example, is unspecific, and specific tools are, for example, for R-type channels, SNX, phytotoxin, or for the capsaicin, we have the capsaicin, which is highly selective, and we can see difference in the patterns showing here. I only want to show you that there's another possibility to have a better view on the activity, on the pattern, on the figure showing here how it looks like. It's helpful for more detailed investigation of the channel. Okay, coming to the dry eye. This is an example of a late state of a dry eye. As here's a red eye with a lot of inflammation and pain and so. And the problem is that the dry eye is with increasing tendency worldwide, 5 to 34%. Germany, we have around 12% with increasing tendency. Symptoms are redness, burning, foreign body sensation. Therapy also, so far, artificial tear substitute, topical anti-inflammation. And the pathogenesis is a disorder tear film production, and cytokines, interleukins, immune cells play a role in that context. There's a so-called circulus vitiosus. The pathogenesis of dry eye is at first instability of the tear film, which can be age-dependent because of malfunction of the myburn gland. This gland is responsible for the tear film, or for the fluid production of that. And then another point is the increased osmolality. Therefore, because there's more salt in the solution than water, and therefore we have an increased osmolality. And this can be due to evaporation, for example, if there's a lot of air in the wind and against the eye and so on. And this can lead to increased osmolality. And therefore, it came in these circulars, it always came to inflammation of the eye surface. And the question, what is the external factor for that point? And the question was first, whether our ion channels, perhaps, that was the first initial question. And indeed, the question is which ion channels and why? And the hypothesis is it should be a channel which can be activated by hypotonic challenge. It should be a channel which is triggering inflammation. It should be a channel which should be, of course, expressed in cornea and conjunctiva cells. And we know, as you know, that TP1 is exactly fulfilling these conditions. That means TP1 can be activated by hypotonic challenge. It can be led to inflammation processes because of cytokine release. And as I showed before, this channel is expressed in cornea and conjunctiva cells. Here, the responsible activation are the hyperosmolality. And the point is, the problem is that we should find a drug which suppresses the TP1 activation, which induce such a problems, and develop the eye drops to suppress this channel. This is the initial idea. Okay, then a summary. I explained you the cornea so that you remember how it looks like inside the eye. I explained you something about the cell lines which we're working, the methods, and also TRPs are partial drug targets, interestingly. And the network is showing here. It's my main collaboration partner is Monika Walting in Dresden. We have a strong collaboration together. Also a DFG project, and this is the internal collaboration within the Charité. Kindly Olaf Strauss, who is the head of the Department of the Expert of Ophthalmology, and Professor Pley, who is a very good expert regarding uveitis in cornea. And this is the collaboration to the endocrinologist, Professor Körle. And also I have a colleague of mine is Fabian Gares, who changed from Erlangen to another city, I don't remember the city. And also my long-lasting colleague, Peter Reinach, who's one of the most, in my view, one leading expert regarding cornea physiology. He's a very good expert, a lot of publications in those. In the meantime, he's retired, but I still have contact. He was in state universities, and he changed to Brazil, to the Sao Paulo University, and he also a lot of connections to the Chinese university. But so far, because of corona problems, he's now still in Brazil. But I have a long-lasting collaboration with him. And these are some of the funders from these projects. Okay, that's the end of my presentation. And I hope that you could enjoy a little bit. And I am curious for your questions. Thank you for your attention.
Thank you, Professor Mergler, for going through that in great detail. I'll go ahead and moderate some of the questions that we received. Did you find the corneal endothelial cells difficult to use on the porta-patch? Was there some assay development required?
It was a moment. It was just a moment. There's something on the door. Sorry, a moment, just. Sorry, and there was someone on the door. Sorry about that.
No problem. It's
Okay, you asked for establishing the planar patch clamping. Initially, I started, of course, with learning classical patch clamp methods, and then I established the Nanion, the small powder patch, and some of the, I forgot her name, she introduced me to using the techniques, and we have just tried the corneal epithelial cells we have prepared, and a certain cell density is necessary so that we can successfully patch these cells, and this was shown. I started planar patch clamp since 2008, and 2010 we had the first large full-length paper showing the chip channels in cornea endothelial cells for the first time, and 2007 we had the first preparation showing the TRPV1 expression, and also some years later we had shown TRPM8 in cornea endothelial cells. A lot of chip channels, as I showed you in some of the slides, so that we could establish it was successfully, and I supervised nearly more than 100 students in lab rotations using the planar patch clamping, also the imaging. It was in most cases successfully, but also, as of course, depending on the cell vitality quality of preparations, a very important factor for having successful recording. So, but altogether, the students are very interested in this interesting technique, which can be easily performed. The most challenge is the understanding about the leak currents and compensation processes and all the physics, which is behind that. And medical students need a little bit more introduction to physics to understand the background of this technique.
And then a follow-up question, how many cells do you get from a preparation? What shape do they have?
Unfortunately, I don't have a photo. The shape is there are different. They have different size. Sometimes for example, for the corneal epithelial cells, as we know, there are different layers on the surface of the eye, and there are smaller cells and larger cells, so that we have different membrane capacity which can be measured. But in general, we also investigate conjunctival cells, so that there are bigger cells, ring cells. These cells have different size, and therefore we used chips with a certain resistance, for example, from 2 to 3.5 mecohm. And for the Neospora, which is like the Toxoplasmosis gondii, we used smaller holes or smaller apertures, resistance around 8 to 10 mecohm, which shows is a smaller hole. That means more appropriate or more suitable for smaller objects. Depending, but in most cases, 2 to 3.5 mecohm worked for the most cell lines. We had no serious problems in that connection.
And then regarding, have you only used acutely isolated primary corneal cells or the cell lines?
Yes, we have checked, of course, freshly preparated primary cell cultures of corneal endothelial cells. We patched them as well and measured the imaging using the calcium imaging, and we could confirm that the electrophysiological properties regarding channel expression, and we did some PCR experiments checking the gene expression of the channels, also functional expression as well, corneal endothelial cells, corneal epithelial cells, and stroma cells as well, and confirming especially the neuroendocrine tumor cells. This was my task in the gastroenterology, to check whether the cell models, for example, one cell line or other insulin-producing cell, one cell line. Also, we checked for the electrophysiological properties and compared them with primary cells, and the data are comparable, meaning that the cell models are properly working in most cases. Of course, there's a limitation, and we need, of course, checking it regularly whether the cell lines are stable. But to my experience, most cell lines, especially the corneal endothelial cells, which also checked by a company regularly for the, for example, the chromosome and DNA properties and so on, at confirming this relative high stability of the cell lines. But we cannot exclude changes because of cell culture conditions, of course, and therefore we we use low cell passage, try to use low cell passage, so we have more safe conditions to avoid changes or genetical changes of the cell lines.
And then to close, do you believe that the increase of incidence of dry eye syndrome worldwide may be due to air quality and pollution? Comment on this?
This is an interesting question. The eye surface is a very sensitive part of the eye, and especially it's the eye surface, as for the ophthalmologists, they investigate if because people who are from other countries, or some people who are doing some journeys to other countries coming back, and then they had some problems with the eye because if you by mistake, if they touch the surface of the finger on the eye surface by mistake, it can lead to infections, and in worst cases it can damage or lead to opacity of the cornea. And there are some there are some therapies available to so that people get back recovery, intact cornea. But this is still very difficult in the kind of diagnosis. It's very difficult to distinguish between fungi, for example, between bacteria or virus. The problem, the eye investigation of the eye surface by eye doctors needs a lot of experience in this matter to distinguish between these different kinds of infections, which are possible, of course. But so far, we try to understand in more details the cornea. So far, there is no artificial cornea available in contrast to retina. There's a project from Tübingen developing a so-called retinal implant project, which was supported by the German government. But so far, it is very difficult. In contrast, in the ear, and the cochlear implantat, which is well established, in contrast to the trying to develop artificial retina or artificial cornea. So far, it's not possible to replace a cornea by artificial cornea. It's not possible. But so far, the only option is to make a cornea transplantation, which is another challenge. The people, although there are no vessels in the cornea, the people need to get some immunosuppressive drugs to avoid possible involvement of the immune system. Therefore, it's a challenge so far, but my goal is to better understand the biology of the specific tissues in the cornea. Each tissue in the cornea has a specific task, so that can fulfill the whole function of the cornea. For example, if there's a problem with the cornea endothelium, the whole cornea can fail in function. That means very sensitive. The density of the endothelial cells is very important. And by the way, recently a student of my group did some scratch assay experiments, meaning making a scratch on the monolayer of stroma cells, and she did a TRPA1 agonist. In comparison to the control experiment, she could show a clearly improved wound healing process after activation of TRPA1. It was an interesting finding, recently presented in a meeting of German Ophthalmology Society, and we are just trying to understand what happens about the cell migration and in connection with role of tip channels. We are just in the beginning to understand the role of tip channels in this context. Very interesting observation regarding wound healing processes. So the cornea stroma is involved in this, or some by accident, some damage of the cornea, which is also related with wound healing processes, so that we can better understand or try to investigate the mechanism behind that.
Perfect. So with that, I think that'll be a good close. Any unanswered questions we can address afterwards when we transcribe this webinar. The next presentation will begin at 2 p.m. Central European Time. Dr. Mark Rogers, Chief Scientific Officer at Nanion Biosciences, will present development and validation of ASIC-1A ligand-gated ion channel drug discovery assays on automated patch clamp platforms. We'd like to thank you, Professor Mergler, for your interesting and exciting presentation. And we welcome our listeners to ask any additional follow-up questions to you or directly to our team here regarding any of the technology used.
Thank you.
Thank you so much. Have a good rest of your day.
Bye-bye.